Okay, so welcome to the Cognition and Natural Sensory Processing Workshop 2021, for short CNSP 2021. So first of all, who are we? So these four people are the organizers. I'm Giovanni from Trinity College Dublin. Then we have Nate, Nick, and Aaron, and you have an opportunity to interact with each of us throughout these 3 days of workshop. So during this workshop, you'll have the opportunities to learn, especially through talks and hands-on tutorials. Our second goal is to connect, and it is to connect researchers and potential collaborators within the CNSP domain. And we have participants from several time zones here. And the third goal is resource sharing. So we are going to share data sets, scripts, guidelines for standardization, and whatever we're going to share with you during this workshop is going to stay online. We're going to maintain it in the next few months in the near future. So who are you instead? As you can see, there is a majority of graduate students and postdoc, which is what we were targeting, and we have also a few faculty members. You can see from this pie chart now that half of you, more or less, were said that they were interested in bringing their own data sets into this workshop and to learn how to use our techniques, our scripts that we're going to share with your own data. So the workshop is about studying cognition and sensory processing in realistic conditions. And so during the workshop, we're going to go through the why we want to do this, how can we do this, and what can we achieve by doing this type of work. And we are going to hear about a recent methodological shift, and we are going to focus on speech and language perception in this particular edition of CNSP 2021. So in the first session, we're going to talk about, if you can see my cursor, we're going to talk about the what and the why of investigating auditory processing with natural sound listening paradigms. In the second session, we're going to talk about the how can we do this, and we will see about encoding and decoding models. And in the third session, instead, we will talk about applied research and case studies. So let's look a little bit more in detail about our schedule so that you have a clear idea of when things will happen. So session one will happen now, of course, and we'll end with a hands-on tutorial that will be relatively brief, mostly to show you where the resources are, how to access them, and so on. The how will be described in session two, and also at the start of day two. So tomorrow there will be a mini session about Python. And at the end of session two, there will be a hands-on tutorial that will be it will go much more in depth than the first one. And finally, the applied research and the start of the mini project, which will be something that you will be doing on your own, or if you want to collaborate with each other. But the main thing is that it's an opportunity for you to go through the code, the scripts that we're going to share, the data sets, and so on. You will come up with questions, with problems, issues that you may have, and we will discuss your questions on day three. So there is about 130, 140 people on this call, or there will be about 140 people. So interaction is going to be mainly through the Q&A Zoom function that you see the button here. And most of you know how this works already. So please ask questions during the talks, and at the end of each talk, the person that is chairing that presentation is going to ask the questions for you. If you feel that you want to ask the question directly and to speak, raise your hand, and we will do our best to enable the microphone for you. There is also a Slack channel, a Slack workspace, that is designed to give you a platform to interact with each other. So the organizers will not be monitoring that during the sessions. And also there is a channel called Questions for Day 3. So whatever question you come up during the mini project, please type them in this channel so that we can collect all the questions, group them, and organize day 3, which will be an opportunity for you finally to interact directly with us and ask questions. So we will schedule it by topic, and so we can communicate directly. And if you have any question, for example, on your particular data set, your particular experiment, we can talk about that. And so please, we're happy to get your feedback at any time. So just type, contact us on Slack, on email, whatever channel. We're going to go through the resource page of our in our website a few times today. So I just wanted to say that we do have this resource page where we have scripts, toolboxes, data sets, and it's meant to stay online for the in the near future. So yeah, so I'll stop sharing now, and this is the start of session 1. So Mick Cross is going to chair this session. So I'll give it to you, Mick.
Thanks, Giovanni. So we're going to get straight into the talks here in session 1. So first up, it's my great pleasure to introduce to you Jonas Obleser. Jonas, who most of you know, did his PhD in psychology at the University of Constance before going to work at the Institute of Cognitive Neuroscience at UCL, and later at the Max Planck Institute of Leipzig, where he established his research group, Auditory Cognition. He's held the chair of physiological psychology at the University of Lübeck since 2016. So his research focuses on the neural dynamics of auditory communication using fMRI, EEG, MEG, computational and psychophysical techniques. His research interests range from processes underlying human cognition and perception to translational aspects of aging to methodological and statistical aspects of neuroscience. So today he's going to talk to us about neural tracking of continuous sensory stimuli. So I'll hand it over to you now, Jonas. You're muted there.
Test, 1, 2, can you hear me? Fantastic. What would be an auditory meeting without somebody having microphone problems, even 1.5 years into this pandemic? I'm not using Zoom too much, I should say. Thanks very much for inviting me. I hope you can hear me. I'm not tracking any of the interaction like hand waving, chat functions, etc. I'm not even seeing this right now. So the organizers have to physically, acoustically stop me, I guess, if I'm going too long or something. So I'm here today to kick us off into this workshop, which is a great honor. And what I'm trying to do, I think, is to set the scene, basically. I try to stay away from too many methodological details. This is, in an essence, a methods workshop, so it's not my job to do the actual methods teaching. I think this will be happening as you go, but I try to do a bit of the framework. The title I picked is actually a title I think I inherited from Giovanni, which is Neural Tracking of Continuous Sensory Stimuli. So let us try and do this. What I want to do is give some introductory remarks, also a bit of a historical perspective, and then in the middle part talk a bit about what, well, my lab does in terms of neural tracking of continuous sensory stimuli, and then maybe send you with some questions that I'm having, problems I'm having into the workshop, and if things go well, those questions might be lurking in the background as you go through the next workshop days. So let me see how I can advance here. Does this work? Yeah. So if we talk about continuous sensory stimuli, I was immediately wondering, what is a continuous sensory stimulus? Like what makes stimuli continuous? And I think of this as a continuum because I think it is a continuum. When we think of a classical psychoacoustic experiment with a lot of silence and a lot of staring at fixation crosses, this is essentially the laboratory classic psychoacoustic condition. It's probably not what Giovanni and colleagues mean when they speak of continuous stimuli, but like where does a continuous stimulation start? If I make those those stimuli more and more rapid and have less and less silence in between. Yes, they get more, the stimulation gets more continuous, but what's really changing then? And I think also in some of the slides of Giovanni, it became obvious that with continuous, we often also mean more naturalistic, more real life, more realistic. But then again, playing an audiobook to a subject or playing a movie to somebody is still not a truly naturalistic, truly ecologically valid stimulation, right? It's still a medium we're working with. So it's an interesting continuum we're moving on. And of course, this will be keeping us busy for the rest of the workshop. Where do you place yourself? Where do you place your research? From a strictly methodological point of view, it's interesting that we're in essence aiming for the same thing. We're aiming to identify how a neural system, here referred to as a linear system, processes input. And if you want to, you could think of the top panel here basically as the classical laboratory experiment. Subject waiting, I play a stimulus, I see what the brain does with it. And the bottom scenario is basically the one we're aiming for, or the one we're currently trying to do. We're having a more or less realistic, continuous, natural stimulus, and we nevertheless try to identify that system. We think this has some advantages, but I will also maybe highlight some of the problems that come with this new approach. I couldn't stop myself from digging a bit into the history of all of this, or I'm personally quite fascinated about the history of some of this development. And when you look into the history, you sooner or later end up with very intriguing techniques. I would say about 50 years ago, there have been already very intriguing setups to study what we would call continuous sensory stimulations. What we're seeing here is a setup by Erich Sutter. I actually don't know much about Erich Sutter. I had a problem unearthing the original paper, but one of my go-to sources here is always Dario Ringa, who writes very gently and very instructively about these topics. And he shows this setup from Erich Sutter from the 1970s, where basically the interesting setup was that you had two tape machines. Two tape machines sharing one tape, essentially. So the tape was going it was a multi-track tape, obviously. So you had one track on that tape playing the stimulus, white noise of sorts here, and then being looped over to the other tape machine, where the brain response was amplified and then recorded. And by basically having a physical distance between the two tape heads, the sound playing tape head and the recording tape head, basically this gave you what we would call nowadays basically the time lag, or the tau, as you will see later in Ed's and others' talk. So if you're interested in that, I really recommend that you look up some of those classics if you want to understand how some of the stuff we're doing today came about. I also find it noteworthy to, not so far in the past, look into how the language and audition fields, the more conventional field of human cognitive neuroscience, has begun incorporating some of the stuff we're doing today. And I wanted to briefly highlight a paper from Olaf Haug, now in Cambridge, who about 15 years ago started also to move away from classic single word linguistic experiments, which was the state of the art at the time, and to a degree still is. You carefully select your stimuli, you control for all kinds of linguistic factors, and then you play those stimuli to your subject, either as audio or on a screen, written. And Olaf basically made the move at the time to say, hey, we can leave the stimulus material, the words, they were still separated words, but we leave them as they are, and we play them to the subject, and we treat, for example, as you see here in those maps, lexical frequency. So how often does a word occur, or the length of a word, a physical feature, or also the semantic coherence between words. we treat those as quasi-continuous regressors. So this moved us into the territory we're in today. And he was doing this, basically, he was running regressions, then at each electrode and at each time sample, and ended up reconstructing something that looked like an ERP, but was essentially a collection of regression weights. At the time, I found this personally quite revealing. Why? Because I was still stuck at the time in the classical laboratory scenario. So to me, the problem we're talking about today basically happened somewhere between 2004 and 2014, I would roughly say. I think those are the 10 years where a lot of stuff happened. This is also not incidentally where Lalor did a lot of his seminal work, and Jonathan Simon. Both are on the call here, and you will hear from Ed later. But from a personal perspective, I was in 2001, 2003, I was still doing my PhD, and I was playing isolated vowels to subjects, was recording auditory evoked magnetic fields, was source localizing them, and I was at the time hunting some form of phonemotopic representation, a phoneme map in the brain or something. Cue 10 years ahead, and you're basically landing somewhere in a Nima Mescarani paper where basically the goal was very comparable. The goal was to identify from brain electrical activity the organizational structure in the superior temporal gyrus of phonemes and phonetic features. So this is just one example where Nima basically shows the whole inventory of phonetic features or sounds in that sense, linguistic sounds, and then shows where maximum sensitivity for these is found. The major difference here is, of course, what's different. We're both using, in a way, linear models. I was using a linear model without really calling it that, or without even knowing properly at the time that I was using a linear model. But Nima basically here has the full natural speech at his hand that he can play to people. And so something happened in between. And today we're basically all, and that's why you're here on the workshop, in the situation that we can play continuous, ongoing speech and unearth brain responses to a whole set of features. So something changed in those 10 years, and we're fortunately on this in this what sounded like the future, to me at least, 10, 15 years ago. Now the interesting thing is, I think the world has always been continuous in a way, right? Sensory stimulation per se, the environment is continuous. Whenever it's not really continuous, it's us making it discontinuous. Certain claims about the neural brain, about the brain notwithstanding, where researchers would think that perception, for example, is discrete. Perception has some element of quantizing, sampling this sensory environment. I think we can agree that the actual environment is continuous. What we do in all the models we will be seeing today or in the workshop, I think, though, is that we break this down. We break always the continuous stimulation down in various ways. You might break it into 200 millisecond chops, you might break it into 20-second chops, but you will always find an element, I think, of breaking it down. Why are you doing this? Essentially because you're training statistical models. You're always optimizing for a certain, there's always a certain function you're optimizing in all your models. You're minimizing a prediction error between something you have observed and something you're modeling from your sensory environment. And ultimately we test our models, and this we also do usually on little chops of this originally continuous world. I think this is just a helpful reminder that what sounds a bit much to start with, or how could I ever model continuous processing? It helps to realize that you're not in the end. You're actually always modeling parts of this or segments of the ongoing sensory environment. Maybe just one word from me. This is really, I'm not the person to speak much about this, but since we have a lot of different backgrounds here on the call, I just wanted to make sure that what I mean by encoding versus decoding models, I've seen that it's a topic on its own in the next session, but for me, it's been quite revealing over the years to realize that encoding versus decoding models basically are two families of research or analysis rationale. It's slightly different questions you're asking here. And I think what we do quite opportunistically is we sometimes ask the one question or we ask the other question, sometimes also for practical reasons. But in essence, there's still different questions. We're answering with the same machinery. That's how I would put it. When we ask an encoding question or are interested in an encoding model, we're basically asking what kind of machinery, what kind of neural algorithm maybe gets me from a given stimulus S, in this case here, the speech ball, a visual stimulus, I'm sorry, to the neural response that I recorded. How do I get there? In this earlier slide that I showed, basically this linear system, this box basically that gets us from a sensory stimulus to a brain response. And a decoding question would be rather, how can I decipher or decode from a given neural response which stimulus was actually present? So this is sort of the classic information decoding question, as it also popped up in functional MRI about 15 years ago, to say we move into questions of decoding. How much information about the sensory world is in our stimulus, is in our neural response that we just recorded. So this is an important distinction, but I think we're essentially using the same model, which is a linear model, which is regression, if you prefer that term, or if it connects better to what you know from your stats undergrad courses. We're always out hunting for sort of a linear operator that gets us from a stimulus to an estimated brain response, or from a recorded brain response to a reconstructed stimulus. So these would be sort of the estimates, that why they have these hats on. So I found that quite revealing to realize that it's essentially the same machinery we're dealing with here. What's happening in my lab around these topics, or what has, let's say, what has kept me busy over the years when it comes to continuous sensory processes? A first realization, I think, that is important to also mention here is when we study the electrophysiological, the neurophysiological brain responses with EEG, MEG, or maybe a local field potential recorded with a sort of an extracellular recording in a piece of cortex. I think it makes sense to distinguish basically two registers or two frequency ranges, probably, or two modes of operation also of the brain. Shown very, very sketchy in orange here on a frequency axis, activity in the delta theta range. So say somewhere between a half a hertz or half cycle per second to up to maybe 8-9 cycles per second, often linked to gamma band activity. This is what Peter Lakatos and Charlie Schroeder about in 2005, 2004 also established as the so-called oscillatory hierarchy, particularly in auditory cortex. There's a connection between those frequency bands. They're sort of nested. But then quite separate from that, and sometimes even contested whether it exists in auditory cortex, I think it does, is the alpha and beta activity. So maybe the activity around 8-12 hertz, and then maybe up to 25 hertz or something. In this range, this is also the range where it can be shown that human brain activity is truly oscillatory, endogenously oscillatory. And making this distinction can be quite helpful in cleaning up your act or your analyses as you go. Why? In part, because they behave very differently. The delta theta range is the frequency range that's very naturally phase locking also to all kinds of incoming sensory stimuli. So if you approach the brain with a continuous auditory stimulus of sorts, this could be speech, this could be music, this could be basically a concatenation of environmental sounds. For one, those stimuli typically have a lot of energy in that frequency range, and the auditory brain, let's put it this way, is very susceptible to these frequencies and phase locks quite precisely to these. And actually, this might be stronger with endogenous amplifiers like attention, but it's happening almost involuntarily, or it is happening involuntarily. These alpha and beta oscillations seem to play a very different role. I do think that they're very interesting, and they can tell us a lot about what a listener actually intends to achieve in a certain listening situation. So the behavioral goals, as I put it here, can be very nicely reflected in alpha beta activity. But and it's also a very important frequency, I think, for a connection between brain areas. So for the interneural communication. However, it's quite hard to actually phase lock. For example, the auditory brain has a tendency to phase lock much less to alpha beta frequencies than it would to delta theta. So we're talking about a different process. And for the sort of models that are relevant to this workshop, so these mainly encoding models, I guess, temporal response functions, you will also note that the focus is often on low frequency activities, say 1 to 10 hertz. And this is where a lot of the phase locking happens. Just briefly mentioning this, this is reflected in the literature, this separation also. You basically have what I would call a speech lineage. You have a lot of people here in this workshop also who are interested in understanding how auditory signals are processed, and they often focus on, I will talk about neural tracking more in a minute, but who focus basically on this slow activity of 1 to 8 hertz and are very interested in phase locked activity. And this alpha beta domain, particularly alpha power, so the amplitude, the magnitude of alpha oscillations, has received a lot of attention in attention research and in memory research coming from the visual domain. And my lab in particular and many others over the years have incorporated that into auditory research. But again, it's a very different, it calls for different analysis tools also. And it's a different sort of research tradition, which we try to bring together, though, on various levels. Maybe to get the nomenclature straight, I should briefly talk about neural tracking versus neural entrainment. It's I apologize to those who've heard me talk over the last 2 years a few times about this, but I wanted to make sure I mention it here. Also, I think for most of the tools you're using in this workshop, it's okay to talk about them as measuring neural entrainment, but I would always add the qualifier, yes, it's neural entrainment in the very broad sense. So it's basically almost like redefining the term entrainment. It's a very broad take on entrainment. Neural entrainment in the narrow sense is a very, I think later on a slide, I call it an epistemologically effortful thing to do. So it's basically, if you want to refer to neural entrainment in the strict or narrow sense, you have to do a lot of extra work to show that you're actually studying neural entrainment. For most of the purposes for people here on this call, I think that's not where they necessarily want to go. They're usually happy with identifying whatever function, whatever filter, whatever kernel gets them from something in the environment to a neural process, a neural signature they're studying. And for that, neural tracking is usually just fine. So basically, what I have not done, what I should do here on this slide is what I refer to as neural tracking is basically neural entrainment in the broad sense. And I will say a bit more about this now. So entrainment in the narrow sense, just very briefly, would be You have an oscillator endogenously active, for example, in auditory cortex. We know that there are probably such processes, delta, theta-like rhythms, or rhythmic changes, I should say, in excitability, shown here in red and blue. This could be, this is a model, what you're seeing here, but this could be, in principle, activity in auditory cortex recorded with an electrode. And entrainment in the narrow sense, in the physicist sense, I should almost say, would be that there is also an oscillator outside, in the physical world, a sensory event happening that is basically also oscillating. This is indicated here by simply by these regular stimulus events happening here on top, these arrows. Entrainment would then be that this endogenous oscillator that has been present before and is present afterwards is temporarily adjusting its phase to these exogenous events. As you can already tell from this little example, claiming that something like this is happening with your sensory stimulus, for example, your speech sound, and your EEG recording or MEG recording, is quite a stretch. It's possible. Some people have shown it. For example, they've shown that such an oscillator keeps ringing, let's call it that, after a stimulation ends. That's a hallmark feature of oscillation, but it's I would say it's expensive to show this, and often people are not successful in doing that. Again, you might have the best chance of showing such a process when you hit an endogenous oscillator. For example, in the auditory range, when you go into the delta and theta range, you have a chance of achieving this, maybe. Colleagues like Christoph Kaiser have made the claim that it's the neural phase, basically. If you are able to, one way or the other, affect the neural phase of a piece of cortex, basically, the endogenous variation of excitation and inhibition, you could be up for an interesting neural coding scheme, basically, because what Christoph shows is that over trials, an endogenous neural oscillation in A1 might be reset essentially to the same phase across events, kind of stereotypically being reset to the same phase. And then what would then follow is basically a stereotypical sequence of excitable and less excitable portions. It's not perfectly sinusoidal, but it's rhythmic enough to also affect then the output of this brain area. So what is shown here in these little graphs, high gain and low gain, would be basically two opposing phases of a slow ongoing oscillation that would have an effect on the actual neural information transmission. Because what we're talking about here would be basically the input-output function of these neurons that are living there in this auditory cortex, and their spike rate would be affected, basically. This is what Christoph shows in this very, very, I highly recommend this J Neuroscience paper by him from 2015 to anybody more interested in what neural phase actually can do to the output, essentially, of a group of neurons. Yeah, very briefly, maybe just interesting, another paper from Christoph from the same year with Stephanie Kaiser together. They showed, they took an interesting approach in messing with continuous speech. That's why I wanted to show this very briefly. Taking this angle of taking an interest in what neural phase of oscillatory activity in auditory cortex can actually do to auditory processing, they messed with the natural pauses that occur in speech. And I find this a bit funny because in these audiobook studies that people like Giovanni and Ed's lab, and also in my lab, we're using a lot. What we usually do is we cut long pauses anyway. They get cut. What Christoph shows here in a way is that the pauses play a role for the speech rhythm. And pauses have so that the silent parts of speech have a characteristic distribution, right? So funk is what you don't play, as the musicians say, right? So the pauses are relevant. But if you cut the pauses, you can basically, so if you try to make the pauses, you jitter the pauses, basically, you make the speech less natural. It's still intelligible, but it's less natural. And this is reflected in the delta phase, in the sense that the mutual information, again, what's common between my sensory stimulus, speech, and my brain response, the mutual information between those two decreases when you do this, when you mess with these pauses. And Christoph also nicely shows here that it's actually the phase that carries the information. So delta phase, always assuming a true oscillatory model, is actually containing information about the stimulus, particularly here in the delta frequency range, as long as the natural quasi-rhythmic structure of speech is somewhat preserved. Again, as I said earlier, this is an expensive claim. It's epistemologically effortful to claim endogenous oscillations, to make sure that it's not just a simple evoked activity change. So for most cases, I would argue, this is actually not what you're after. You're just, it's fine if you don't make strong claims about whether it's a true oscillator or not. And when you do this, you're basically more liberal, and you would call it, you can call it neural entrainment if you want to, but it's then entrainment in the broad sense, I would think. A physicist would object that you've actually shown entrainment, but it's still fine, I think, to call it neural tracking, because what we will be doing for the rest of the workshop is we will be looking into what kind of physical features can be reconstructed from neural features. Basically, we're looking for whatever link function that gets us, basically, or whatever linear operator gets us from features of the stimulus to features in the brain. So this is really the goal of neural tracking. You try to go from something in the environment to something in the brain. I put it so generally because I think this is the real power of this. The power of the system lies in its generality. And you see, basically, we all witness every month somebody picking up on a new physical feature that or a linguistic feature, for that matter, that they're tracking in the brain response. And what you try to do is you try to identify basically this function, that this kernel, this filter that gets you from the environment to the recorded brain response, or in a decoding model, vice versa. In our work, and I just wanted to, only data point I wanted to briefly show, in our work, we're basically we're playing the same game. We're using neural tracking as a measure to study auditory attention. And we've recently done this in a very, very large sample of about almost 160 people of varying age, rather on the older end, rather like older listeners who are thought to have problems in cocktail party listening situations. And we studied what neural tracking can actually tell us about success at the cocktail party. So shown here in orange, we try to model behavior. We try to model how well you're doing in a challenging cocktail party attentive listening situation. And we studied neural tracking, but we also studied alpha power, which I'm not talking much about today. But this is an interesting study, and I can only point you to the paper came out last week, or this week actually, yesterday, I think. No, last week. Last week it came out. Can only point you to this rich paper because it shows that on this continuum that we're talking about, from a trial-like laboratory trial structure to a continuous ongoing speech, we're rather here on the trial end. We used 2-second sentences, presented those 2 at a time, so dichotic stimulation, and measured behavioral responses to words in the sentence. So rather a laboratory experiment, but we nevertheless used neural tracking for that. We were able to show that neural tracking also, when restricted to a trial-like structure, 2-second sentences, is able to predict behavior on a between subjects level. So those people who show stronger neural tracking differentiation between what they're supposed to track, attend, versus what they're supposed to ignore, those people who do this more are also the faster responders. And also, I think more interesting, and actually the more robust finding in those data is the fact that so it's more robust because it replicates better, is what's shown here on the left side. Namely that across trials, there are trials where you do more of a neural tracking attention differentiation and sometimes less. And when you do this more, you're actually more likely to get this sentence correct. So I think this is a nice proof that neural tracking, as established normally in rather audiobook listening scenarios, so continuous stimuli, also works pretty well on a single trial level. Overall predictive accuracy, so the goodness of fit essentially of the model is not as good, obviously, when you have shorter segments, but it works surprisingly well, and we can use it to answer cognitive neuroscience questions with it. How am I doing time-wise, Giovanni? Do I have a few more minutes? Or Mick, sorry.
Yeah, even about another 8 minutes.
Okay, cool. So to sum up, basically, I wanted to sort of share my enthusiasm about the neural tracking framework, partly because I think with the same mathematical models, we're nowadays seeing many different neural signals being modeled, basically, right? So what is drawn here as an MEG-EEG signal could be a lot of different things. Nima Meskarani and others, or Kirill Nursky, they're basically using the power envelope, so the magnitude changes in very high frequency activity, as recorded from various locations in STG and auditory cortex. In my lab and in the lab by, I don't know, Elia Formisano, for example, very often it's a bold time course. So you're using fMRI and you're recording activity from all over the brain, from certain voxels, and you're treating those slow, bold changes as your dependent measure that you're modeling. As I briefly alluded to in some work by Christoph Kaiser and many of our colleagues working invasively in animal recordings, spike trains, or sometimes also instantaneous, so interpolated spike rate, basically instantaneous estimates of the firing rate, can be interesting time courses to be modeled that way. And also power envelopes in this alpha beta band that I talked about. That doesn't entrain very well, but nevertheless, you might very well be interested in the fluctuations in alpha beta band. And under the same umbrella, you basically can now turn to the stimulus side, to the continuous sensory stimulation side. And this is what I meant earlier, where people are very, very creatively coming up with new features, basically, of this physical world that can be modeled in this context. So on the stimulus side, acoustic features in various complexity and various levels of modeling are an obvious target for these kinds of analyses. We're seeing a lot of activity for linguistic events, so word onsets, certain phrase boundaries, also on a phonological level or phonetic level, work by Giovanni, of course, very prominently a few years ago, and generally Ed's lab is, of course, very active in this field also. What we've recently done is we've trained a neural network, basically long short-term memory networks with various layers, and trained networks to do predictions in speech, and then took basically the output from those layers into such a encoding decoding framework, and we're looking for how voxels in the brain actually track these layer specific predictive markers from the networks, and also once you're doing all that, it's a small step to also incorporate behavior or movement, I would say, in general. So incorporating camera data, recordings, like there's a fantastic paper from Anne Churchland's lab from 1.5 years ago in Nature Neuroscience that I very much liked in this sense. They used basically, they did a PCA on the camera data that they had, the video recordings of a mouse, basically. And then the whisker movements, the head movements can become regressors in such a framework. So where do we stand with all of this? We're basically here hitting a continuum, as I said, a continuous stimuli continuum, but there are choices to be made. As you place yourself with your study somewhere on that continuum, you're basically inevitably take choices regarding how ecologically valid you want your study. What kind of behavior are you getting out of this? And also sometimes overlooked a bit, as we move into more natural territory, we also have more covariation or more basically mutual dependencies between certain features in the audio, in the visual stimuli that occur. And this affects, of course, your ability to model the brain response. So the three problems I want to leave you with and send you into the workshop are basically therefore what I would call the behavioral problem, the information problem, and the dimensionality problem. Very briefly, the behavioral problem is, I think, maybe obvious. As you move to continuous stimuli, you're losing first the ability, the obvious ability to get very, very frequent behavioral reports from your subject. You don't get response times every trial. You don't get accuracy every trial. So how do we make up for this? This is an open question with a lot of ideas. In my lab, shown here, we're currently trying to place little repeats in the signal as we go. So you listen to your audiobook, like in most Lalor Simon study, you would be listening to audiobooks, you try to comprehend, but it's almost like a dual task thing, because at the same time, you're tracking target events and you can do psychophysics on this. This is one way of answering that problem, but it's not the final answer, I think. So I think we are in need of new quasi-continuous behavioral responses. And this is maybe a task for all of you out there. The information problem, basically what I mean by that is to say, our machinery is very focused on covariation, covariance, recovery, explaining variance. You can also call it mutual information. But simply because you're having mutual information between something in the brain and something in the stimulus does not necessarily explain to you how this thing works. It's maybe an obvious problem. I will not go for reasons of time into this little neat example from Eric Jonas and Konrad Körding. It's a very provocative paper, but it sort of touches upon these problems that I'm highlighting here. I'd rather leave you with this quote from David et al., who said, like, we could ask more generally, they say, whether the physical differences that we measure and that we see in the brain responses, are they actually making a difference for further processes in the brain, right? So only because you can decode information from auditory cortex, does that mean that this information is relevant for the ensuing downstream neural processes? This is not a trivial problem, and that should keep everybody on their toes who works and relies on information and recovering variance. And the last problem, and then it's really my last slide, is the so-called dimensionality problem. What's the right way of modeling EEG, MEG data, basically, in order to do justice to those data? What I show here a bit as a continuum, so our MEG, EEG signals could be analyzed in various ways. I think when we analyze a single channel or a single voxel, we might be, basically, we're missing something. We're doing univariate analyses, hundreds of them maybe, or maybe sometimes thousands in fMRI, but they're not really independent, are they? I mean, we treat them as univariate problems, but they're obviously not. On the other hand, only because we have, say, 300 channels, or only because we have 100,000 voxels, does not mean that the data is inherently 100,000 dimensional, obviously. So we can also sometimes have artificially high dimensionality. And in that sense, I think it's worth lurking over to what's happening in fMRI research. And there's been an interesting paper by Max Shine recently, where they basically showed that functional MRI data is nicely captured. Basically, something like 5 spatiotemporal components that you can then think of brain activity basically as activity moving around in a, say, 5-dimensional state space, right? So it's shown here as a 3-dimensional thing. And I think this is a direction I would like the field to go a bit into, to say we model also EEG, MEG activity basically in a rather modestly dimensional space, and can then sort of apply our encoding models there. But this is really just an idea I wanted to leave you with. So the story continues somewhere between naturalistic, continuous stimuli and laboratory control and close monitoring of behavior. And you're in the middle of it. And I wish you a good time here at the workshop. Thank you very much. Thanks to the people who do the actual work in my lab. And thanks to you.
Thanks very much, Jonas. That was brilliant. So I'm going to open up to questions now, which you can put into the Q&A box, which you can access there at the bottom of Zoom. We have a question here from Talat. They asked, how do you characterize the long pauses during audiobook experiments? How do you determine pauses greater than 250 to 300 milliseconds should be removed? My question is from brain activity standpoint or from attentional standpoint?
So I understand the question. I don't know how you understand the question, Mick, but I understand the question to basically be like very practical about how to go about, like where do you do the cutoff? And I think there are various ways of doing this. I mean, so the decision by others in the literature, people like Naiding or Jonathan, has been to say, okay, I draw a cutoff somewhere at 300, 400 milliseconds. But from a speech rhythm point of view, as I have tried to show with this data from Christoph, this is of course also doing something to the natural soundingness of your stimulus. These pauses are important. These pauses might be actually what helps a listener also to sort of stay engaged. I mean, are you really ruining the process you're trying to study? That's a different question. But generally speaking, if you really want to go about this, you would probably need to run sort of almost like pilot experiments to make sure on a perceptual level. I think if one goal of you is to be ecologically valid, then you should take some care not to ruin this ecological validity by just removing pauses. We might be removing those pauses for boring mathematical reasons, basically, not to have regressor data instead of regressor silence. But it's an open question, I think, actually, what this actually does. And Christoph's paper is an interesting hint. I mean, what I didn't show you is that, and that's also, it's important actually, because Christoph shows in the paper, the intelligibility of the signal suffers not at all from removing those breaks. But the neural phase and the mutual information with the phase goes down a bit. But it wasn't high to start with, right? Neural phase doesn't explain too much of the variance in the stimulus anyway. So it's a tricky question. Thanks for that.
Just as a follow-up, how do you think then, so if you were to mess around with your stimuli a bit in order to try and increase the information rate and, you know, shorten your experimental time, how do you think that would impact different groups differently? So say if you were studying older aged participants, do you think that could have even more of an impact and maybe somehow bias your results?
That's a very interesting question. I think it touches a bit upon the inherent neural dynamics. I think so in the working groups here on the call today, like in Ed's lab and also in my lab, we've been, for practical reason, very interested in this with how you're always wondering with how few data do you get away and do you arrive at the same conclusion? So that's a statistical question, just basically. But if the inherent dynamics of, say, tuning in to speech is different in older listeners than it is in younger, or if it depends on sort of state variables like vigilance or something, then this might very well have an impact, and we might be treating subjects differently without intending to, by giving them longer or shorter speech segments to work with, for example. Again, I think an open question, actually one that could be nicely addressed here in this workshop also. Well, you don't have older and younger data maybe, but just in principle, I think it's an open question what this would do to different groups. And I also don't have a very strong indication that it would do anything, to be honest.
Thanks for that. Amir asked, why are we focusing on linear models? Why not non-linear models such as deep networks? That's a big discussion.
Well, that's a very good point. I mean, I hope what I intended to do, I didn't do justice to deep neural networks or something here. But what I aim to show is that the framework to me is still the same, basically. So what you would be doing is you would be replacing that. Basically, there are different ways of arriving at this transfer function, or at this kernel. Basically, I think a linear. It's always parsimonious to work with a linear model if you can, because it's it's readily intelligible to you as a researcher. It's easy to see what it does. I'm not a super huge fan, generally speaking, just for say if you would employ a deep neural network here or something to do that, you can do this nicely to arrive at maybe higher accuracies or at comparable accuracies. But there's a lot of additional time you would be spending unpacking what the network has been doing. So it's okay to be excited about deep neural networks, but I think it's also a researcher's duty to pick the most simple tool that gets you somewhere. So I mean, it's a bit more a fun comment on the side, but I think deep neural networks should basically, you shouldn't just use R-square in the sense of explain variance, but you should really put that in relation to the training data or to the carbon footprint or whatever they're doing. And I think the linear model might very often still then win. That's why so many people from the linear modeling field are so get so angry or annoyed when people, I don't know, basically do things with a deep neural network that you could also do with a simple logistic linear regression sometimes.
Thanks for that. Right, well, if no one has any other questions, we'll continue on to the next session in 2 minutes. You can access all of the questions. Thanks very much, Jonas, for that.
Thank you.
You can access all of the questions in the Q&A box, and we've had a few nice answers as well typed in there as well. Okay, so next up, it's my pleasure to introduce our second speaker, Ed Lalor, who at some point has supervised each of the organizers of the workshop. So I guess he's to blame for all of this. Ed, who a lot of you know, did his PhD in biomedical engineering at UCD and his postdoctoral research at the Nathan Klein Institute for Psychiatric Research, Trinity College, and briefly at UCL as a Marie Curie Fellow before taking the faculty position at Trinity. In 2016, he joined the Department of Biomedical Engineering and Neuroscience at the University of Rochester, New York, where he's currently an associate professor. So Ed's work has focused on developing novel methodologies for neuroscience research and the application of those methodologies to study human sensory perceptual and cognitive processing in health and disease. His work has led to improved understanding of the neurophysiology of receptive speech processing and how it's affected by attention and multisensory input, and some novel insights into neurophysiology of early sensory deficits and disorders such as autism and schizophrenia. So today Ed's going to be talking to us about the temporal response function, its concept and basic research. So I'm going to hand it over to Ed now. Thanks.
Thanks, Mick, for the introduction and for inviting me to contribute to your guys' workshop. And thanks, Jonas, for a great talk, as always. I thought I'm hoping my follow-on does a reasonable job of actually hitting on some of the nice ideas that Jonas raised in his talk. He did so in his characteristically, more philosophically kind of thoughtful manner. I'm going to do it in a slightly more didactic and specific way with examples to specific data sets and so on. But I hope to hit some of the same ideas that Jonas raised in his initial talk. And I've been instructed not to bog down in the weeds too much with methodology, because there's lots of time in subsequent sessions, I think, for getting into the weeds on methodological details. So with that in mind, again, I'll kind of three almost apologies at the outset of my talk. The first is that it will be quite didactic. The idea is to try to give kind of very instructive insights or introduction to some of the ideas that are going to be showing up today and tomorrow. Some of that means that there's a relative lack of new ideas and new things necessarily in the talk. So apologies to some folks who've heard me deliver talks in the past, that you're going to be seeing a lot of the same stuff again. But again, the point was to serve as an introduction to some of the ideas. A second apology quickly, just to say that the talk is overwhelmingly focused on some of our own previous research. And that's just for the boring reason that, it's easier for me to lay my hands on the figures to make the points that I want to make quickly from some of our own work. So apologies in advance to a relative lack of citation of all the other great work that's going on by groups like Jonas himself and Jonathan Simon and many others that are doing similar, using similar approaches to tackle very interesting questions. And then the last caveat is that I may have tried to cover too much here. So what I'll end up likely doing is moving quite quickly and again skimming over details in the hope of hitting some kind of overarching conceptual ideas, just to get folks thinking about these things, and we'll drill into the details of those, I assume, in the next talks later today and tomorrow. So I'll share my slides here and kick off. So, stop sharing. I'm not sure if that worked. I'll try again. Mick, are you guys seeing a full screen thing there now?
Yeah, that's perfect.
Okay, great. Okay, so just a quick introduction to the temporal response function itself. And again, following a lot of what Jonas said, the idea here is, and I'm going to be kind of agnostic about what kind of stimulus we're talking about for the time being, but let's just imagine any stimulus. This could be, in this case, it happens to be a speech signal, and the red line happens to be the temporal, the amplitude envelope of that speech. But this could have been, the luminance of a movie scene in a video, or the strength of vibrations of a stimulus placed on your finger, or whatever. The point is that it's just a time varying stimulus feature that we can characterize with numbers that change over time. And meanwhile, we're recording some data. It could be EEG, MEG, electrocorticography, whatever. And then the point is of temporal response functions is really just to ask the question, how does this changes in this stimulus feature show up in my EEG data or neural data? And how can I model that with some mathematical function in the middle that captures how this X turns into all of these different Ys? And there's many different ways to do this. The temporal response function in particular does this using just an assumption of linearity, as was coming up in the chat there. Why are we doing linearity? Well, it does. We use linearity because it's relatively simple, relatively easy to fit a linear model with limited amounts of data, which is always an issue. EEG is not actually all that high dimensional, right? You've got 128 channels and lots of time points, but each channel is quite highly correlated with its neighbor. And each time point is quite highly correlated with the next time point. So the data itself are not actually all that high dimensional. So fitting big, complicated deep neural networks with relatively low dimensional data like this is not that easy to do without overfitting the model. So for those reasons, in terms of interpretability and the actual, practical issue of fitting a model, a robust model with limited amounts of data, linear models have been a mainstay of neuroscience research for decades. And so this is what we do. We end up with something that looks a little bit like a classic event-related potential. And again, Jonas showed us these nice impulse response plots in his talk that kind of explains why that is. It should look a bit like an ERP because, you know, if I was to send in an impulse here on the left, I would get an impulse out, or I would get an impulse response out that would look like an ERP. And so here I'm kind of deriving that impulse response function or temporal response function, but I'm doing so using a more continuous stimulus and linear regression. The beauty of this as well in terms of usability is that we can then piggyback on the kind of wealth of tools that have been developed over decades for ERP research using kind of classic event-related potentials. Folks have learned how to, you know, develop interesting source models to try to estimate where sounds are coming from in the brain, where responses are coming from in the brain. And so you can do that with temporal response functions, again, leveraging the kind of existing methods out there that were developed originally for event-related potentials. You can use those on temporal response functions. So it's a kind of it's a natural, you know, relatively easy transition from you know, discrete beeps and clicks type experiments into more complex continuous stimuli, while still being able to use a lot of the tools that were originally developed for those event-related potentials. But it can be used in other ways also, and again, this will be a theme that will come up in later talks, I'm sure, and in the workshop hands-on sessions. It's, you know, it's not just the temporal response function itself that acts as a dependent measure here. You can look at this, of course, and see how it's distributed on the head, see where the peaks and troughs are, at what latencies, what amplitudes, and so on. But you can also think of it as a model framework, as a so-called encoding model, where, you know, if you've got, say, for example, 10 trials of an experiment, you can fit one of these temporal response functions using, say, 9 trials, and then try to predict the data from the 10th trial using this model. And so this is a slightly different, you know, reworking of the same framework, but it gives you now a new dependent measure. Your new dependent measure is the ability to actually predict responses to new stimuli. And in fact, you know, in some ways you could argue, and it has been argued quite nicely, a paper by one of my former mentors, Matteo Carandini, who argued that, you know, we can actually properly say that we know what a sensory system does when we can accurately predict the response to any stimulus that the subject might experience. Now, we're not there and may never get there, but the idea here is that one can use this prediction accuracy of new data as a measure, dependent measure, to say that you've successfully captured something to do with the sensory processing of that stimulus. So it's the same framework, but just used in a slightly different way. And then finally, and you can see again on the head where it is on the head that you can predict good responses and where you can't. This is an auditory response as it happens. And so you can see that you're doing a good job of predicting EEG channels where auditory responses would be expected to be, and you're not predicting data over occipital cortex very well, for example, which makes sense in this case. And then you can use it also as a decoder. And again, I won't get into the details too much because future talks are going to do that. But the idea here is to, and something that Jonas touched on in his talk, rather than trying to predict each channel one by one, which is, you know, treating each of these signals like a separate, entirely independent thing to be predicted, when in fact the data actually are all quite closely related across channels, many of the channels. You know, this is quite suboptimal to predict one channel at a time and ignore the fact that the data across channels is related. By using one of these decoding frameworks, where you essentially attempt to reconstruct an estimate of the stimulus feature from the data. So you're going the opposite direction. You've got the data and you're trying to say, well, what was the stimulus that caused these data in the first place? This has a major advantage in taking in all of the channels at the same time into a multivariate model that has a prediction accuracy of the reconstructed stimulus now as its dependent measure. And the major advantage of it is that, again, by taking all the channels at the same time, you get to exploit the common signal across channels. You get to be sensitive to the noise that's different on different channels and factor that out of your analysis in a slightly more powerful way. Again, you get different weightings on different channels that can be informative as you do this. And again, this is an auditory response. So you can see the channels over temporal scalp are very important here in reconstructing this auditory stimulus. And so again, you end up with a dependent measure that is this kind of reconstruction accuracy, and a dependent measure that's the weight of of the weights that are being applied to the different channels at different time points as dependent measures. It has this major advantage, and I'll show that later with some example of a particular experiment, a major advantage in better exploiting the available data so that you can massively increase your sensitivity to brain responses to sensory stimuli. And it really changed the way our lab does business in many respects by providing us with a much greater sensitivity to stimulus effects than we had seen with our forward models. However, we have struggled in many instances in terms of what it is that we can reconstruct from EEG. So again, I'll highlight a few examples there as we move forward of ways in which forward models can be used to test hypotheses about how different stimulus features are processed by the brain, but how decoding those features can actually be quite tricky with EEG and with TRFs. Okay, so in terms of, again, my didactic talk here to try to set the scene, and again, hitting on some of the points that Jonas did, my plan is to go through this framework here using a series of examples to highlight that what we're talking about here, again, is looking at the question of how specific stimulus features show up in the data. So it really does, an awful lot depends. I mean, it nearly all depends on what you mean here by your stimulus and your stimulus feature. What is it that you're actually interested in asking of your data? What is this X that you care about so much? But it also depends on what you mean by the data. There are, and again, Jonas touched on this briefly, the idea that different EEG features, the phase of low frequencies versus alpha power bands, for example, you had that nice plot with the different frequency bands and what they may reflect. So again, that's very important as to what it is that you care about and how to fully exploit your data, depending on what you're interested in. It's not just send in the raw EEG all the time. That makes sense. And then also I'll spend a couple of minutes talking about the TRF itself in the middle here, where we've made some linear assumptions that are not ideal, and where in our hands, at least, the TRF has failed to sufficiently help us in answering certain questions, and we've had to actually rely on related methods. So I'll just very briefly touch on some of those other methods and how the TRF relates to them. Okay, so we'll start off by talking about this X. What is this X, and what, how does that influence the kind of TRFs one might actually arrive at? And I'm going to go back in time a little bit to some of the earliest work that I did on this topic as a student, which was actually in the visual system. So rather than thinking automatically about speech, which is a lot of the stuff we've done in recent years, just to take a completely different look at something that folks may not have seen before. So when I originally started doing this, I started doing it with visual stimuli, these checkerboard patterns where I would modulate the luminance or contrast of the checkerboard, like every refresh of the monitor. So we had a checkerboard pattern that was kind of very rapidly changing luminance or contrast on the screen. And the idea here was that the X in our analysis, the stimulus feature we cared about, was that luminance or that contrast of the stimulus. And so the question we ask here when we derive a TRF using this stimulus contrast was, show me how the EEG changes as a function of the stimulus contrast. That's the question that this analysis asks. Just by way of anecdote, in these days, those old days, we call the TRF the Vespa, as a kind of a feeble enough attempt to get the new method to catch on by giving it a kind of a snazzy, what we thought of as a snazzy name. It was one of those names that you think of first and then have to reverse engineer the actual acronym by coming up with, I think it was Visual Evoked Spread Spectrum Analysis or something that we made it stand for. But in any case, when we mapped onto the auditory system a few years later and started calling it the ASPA, and nobody knew how to pronounce that, and other labs were using very similar techniques and had been for a long time, so we realized we had to just give up the game on giving these things fancy names and started to call it the temporal response function. So anywhere you see Vespa, I really mean the temporal response function, the visual temporal response function. So here again, the question was, how does the data change as a function of the luminance or the contrast of the stimulus? And we wanted to compare that to the kind of classic ERP, the classic visual evoked potential that you can get if you just take a checkerboard, for example, and pop it on the screen, or you take a checkerboard and quickly reverse it back to front and again. So when you do that and you actually look at the data on the back of the head, a channel at the middle of channel OZ over the middle occipital scalp, the VESPA and the VEP look really quite similar. And again, makes sense. One of them, the VEP is being derived, it's an impulse response. You're hitting the system with a checkerboard reversal, which is an impulse, and producing an impulse response. And the VESPA is an impulse response function in a sense. You're sending in a dynamically changing stimulus, but then you're asking how does that stimulus produce the data at the output in a linear way. So you end up with a linear impulse response. So it kind of makes sense that they look similar. But interestingly, and this is important for thinking about TRFs in general, they're not the same in some important ways. So for example, if you look at a VESPA, a visual TRF that again was evoked based on asking the question, how does the data change as a function of the contrast of the visual stimulus? You end up with a response that kicks off very early in the very center of the occipital kind of scalp here at around 75 milliseconds. And then it stays right in the middle of the occipital scalp as it flips from negative to positive, and then it kind of just disappears. Meanwhile, for a visual evoked potential, again, simply a checkerboard that reverses, you get a very early response right over the center of the occipital scalp, just like the visual TRF. But then it spreads all across the back of the scalp. And in fact, it seems to almost divide and propagate away from the center here down towards kind of lower temporal areas as it progresses. So really very different topographies, despite the fact that if you just look at the channel in the middle, they look kind of similar. So what's going on here? Again, I've inserted a little quick anecdote here because I was a young, not that young, but a younger student, an early stage postdoc when I was doing some of this. And we got very excited that this actually could be very useful at discriminating patients with schizophrenia from healthy controls. And the reason was that VEP studies had frequently shown that visual evoked potentials for patients with schizophrenia were much smaller than those were for healthy controls. Now, some patients had nice big VEPs and some controls had very small VEPs, but on average, there was this big difference. However, it wasn't useful diagnostically or in a clinic because, again, like I say, some patients had big responses and some controls had small ones. So all we got was this kind of average group difference. And we felt that this visual TRF, this VESPA, was going to be able to actually separate out the groups much better. And I got super excited. I was young and naive and wrote a patent application and thought, not only am I going to have a big impact on the well-being of patients with schizophrenia in terms of helping with diagnosis, but I might also make lots of money with my patent and all sorts of stuff. And so we ran the experiment to see how the VESPA would look between these two groups, expecting a nice big response for healthy controls and a flat line for patients. I won't go into the reasons why we expected that, but that's what we expected. And this is what actually happened. We found that there was zero difference in the TRF between these controls and the patients. Same group of controls and patients here, which was very disheartening for me for about 24 hours until I kind of realized this was very interesting. Why does a checkerboard that reverses produce a big difference and a checkerboard that just flickers does not produce a big difference? So scientifically, this was a very interesting question. And again, it gets to the idea of what is this X we're talking about that we've modulated? The visual system is a very complicated thing. There's a whole ton of different visual areas in the brain that all are specialized for processing different features of a stimulus. Fusiform face area that cares about faces, parahippocampal place area that cares about places that you've seen that you might be familiar with, or buildings or whatever, motion sensitive areas, object sensitive areas, etc. And it turns out that a lot of these areas don't care too much about contrast. I mean, they in other words, well, they care a little bit that, let me take an object processing area like the lateral occipital area. This area cares about things like if I'm seeing a coffee mug versus a cell phone. Once the object is illuminated well enough to see it, and I make it brighter and brighter and brighter, that object no longer cares about the luminance of the object. Once it can see that it's a coffee mug, it will fire. Make the coffee mug brighter, it'll fire just the same. And don't forget this, the question we were asking with our data was, how does the EEG change as a function of changing the contrast of this visual checkerboard? it turns out all of these areas, LO, FFA, these higher order areas in the visual hierarchy, they don't care about the checkerboard changing anymore. Once they see that it's a checkerboard, they don't care if it's a bright checkerboard versus a relatively gray checkerboard, a lower contrast checkerboard. And this explains why the visual TRF versus the VEP was so focused over the midline occipital scalp and never moved. It's because the only areas that actually care about contrast are these early areas like V1 and V2 that are located right at the occipital kind of pole here. With a VEP, when you whack the system with a new stimulus, like a checkerboard that just pops on the screen, that information floods across many of these different visual areas to give you a much broader response across the scalp. So the X that you send in and the specific question that you ask of the data is going to very, very strongly determine the TRF that you actually get. There is no such thing as the TRF. The TRF is a method for asking that question. Show me how the data change as a function of changes in this particular stimulus feature that I'm interested in. To bring that point just home a bit further, and inspired by this effort to try to find a EEG measure that might discriminate patients with schizophrenia from those who don't, we followed this up a few years ago by trying to make a new stimulus that would derive from areas up the so-called dorsal visual stream. Doesn't really matter the details, but we had reason to believe that data from patients might be interesting to look at up in these visual areas that care about things like motion and visual flow and things like that. So instead of making a checkerboard, or should I say, as well as making a checkerboard that changed in contrast, we made this stimulus that was made up of quite large dots that were spread all across quite a large monitor and that were moving around either randomly or coherently. So sometimes all the dots will be moving together, sometimes half the dots will be moving together, sometimes all the dots will be moving in random directions. And again, here now we're asking the question, show me how the data change as a function of the coherence of the motion in the stimulus. Now V1, early visual areas that are generating these responses, V1 can't see the coherent motion. The dots are too big. The cells in V1 only see a small part of the world. They cannot tell that the dots are moving together or separately or whatever. They just see dots moving. Each cell cannot see more than one dot. And so as a result, we ended up with this interesting response that was quite a bit later. We didn't see anything kicking off at 75 milliseconds. It wasn't really starting till around 100 or 90 or 100 milliseconds. And it came from much further up the scalp. For those folks who are not used to EEG data, this is quite a significant difference here. You've got the contrast response coming from very early visual areas, and you've got a motion response coming from way up the dorsal stream, which is exactly what we were aiming for. So again, this is just by way of saying, same analysis, different stimuli, and most importantly, different X's. This X going into the analysis is different. It's a different stimulus feature, and we end up with very different TRFs because we're asking a different question. Okay. One of the major themes of this workshop obviously is natural stimuli, or more natural stimuli, to not overstate it, Jonas, such as speech from things like audiobooks and movies and so on. That's something that we've spent a lot of time on, a lot of groups are interested in. And so how does this idea of this hierarchy that I've just talked about map onto this question of speech? Well, there's a lot of work to show that the speech processing is carried out by a hierarchical network. And that in fact, you've got brain areas such as in green here that are, early auditory regions that are performing kind of acoustic spectral temporal analysis of the speech signal. But then this moves on to higher order areas that start to care about speech specific sounds, the phonology of the actual speech that we hear. That passes on information to brain areas that might care about the actual words that we're hearing and recognizing those words as things that we're familiar with, all the way up to entire swathes of cortex that have been shown to actually care about the meaning of the words and what they mean. So we have a very, much like the visual system, we have a very hierarchically organized auditory system. So if you're playing speech at somebody and then trying to derive a TRF, what does that TRF mean? Well, it very much depends on what you fed in as this X. What is it about the speech that you care about? That's very much going to determine what the TRF looks like, and honestly, where the TRF is coming from? What are, what brain areas are generating the activity that is producing this TRF? It depends on the feature that you've actually asked about. And so to demonstrate that, we've done several papers over the years trying to explore beyond, for example, looking at speech envelopes by asking questions about how EEG data reflect not just the speech envelope, but the full acoustic kind of spectrogram of the speech versus the actual kind of phonemes and phonetic features of speech. These are different things, because, if I were a robot and I said the same kind of phonemes the same way every time, if I said ah the same exact way every time, then a spectrogram representation of a speech signal and a phoneme representation of a speech signal would basically be identical. One would essentially be shorthand for the other. But I'm not a robot, and I sometimes say ah, and I sometimes say ah, and I sometimes whisper ah. So all the time I say that, and you're listening to me, you will understand that you will hear ah and recognize it as an ah, even though the actual acoustics of it are different. So one might then expect that if I were to try to use this spectrogram here as my X, then I'm asking the question, show me how the data change as a function of the different frequencies and over time that you've heard. One might expect that this derives from brain areas that care about all those little spectrotemporal differences. So early acoustic areas that just care about the actual sounds and don't even necessarily care that it's speech that you're hearing, just care about the fact that it's frequencies that change over time. Meanwhile, if I actually say, show me how the data change as a function of these kind of categorical speech units, like ahs, that might sound different from time to time, but they're still an ah. Does the EEG reflect that kind of categorical nature of those phonemes? You might expect that it should, because we kind of perceive speech categorically. Again, I could say the word, a window, or I could say window, and they sound different, but you recognize them as the same word both times. So again, this is something we were interested in exploring, using this idea that different neurons lower in the hierarchy might just care about the raw sounds versus neural populations that are further up this hierarchy that Hickok and Popple have talked about, and others, Sophie Scott and many others have talked about. One might expect that whatever X you put into your analysis might have different effects on the TRF that you ultimately derive. So to test this idea, we actually, assume that we're getting contributions to our EEG from low-level areas that care about the different sounds, as well as higher-level neuronal populations that care about the fact that you are recognizing categorical units of speech like phonemes. And when you do this, we use a prediction kind of modeling framework, a forward encoding modeling framework to do this. We found that when you try to predict EEG using both the acoustic information and the categorical phonemes, that you do a better job of predicting the EEG. So in our minds, this was evidence that the EEG reflected both the acoustic processing as well as a higher level kind of categorical processing of linguistic units. That makes sense. And a lot of this work was inspired by Nima Meskarani and Eddie Chang, some of their works that again, Jonas referred to in his talk, particularly their paper in Science magazine in 2014. We tested this using speech that was played backwards, literally reversed in time, so it doesn't really sound like recognizable speech anymore, and found that the phonemes in this case don't add any value because you can't recognize them anymore, and so on. More recently, we've built on this again, same data, but asked a different question. We've been inspired by work from Alexander Huth, who's been working with Jack Gallant for years, exploring, and many other groups are doing this these days as well, Stefan Frank, for example, looking at trying to quantify the meaning of words in a semantic sense. There are different ways to do this. I always give the same example when I'm kind of talking about the meaning of words. I give the example, if I say something like, the dentist told me to brush my teeth. When I say the word teeth there, I gave you effectively no new information, because it was so obvious when I said the dentist told me to brush my, it was so overwhelmingly likely that I was going to say teeth, that when you heard teeth, you weren't surprised, you didn't really learn anything new, you knew it was coming. So the amount of information content in the word teeth there is very low. And so you can quantify the amount of information in words based on their context. You can relate words in a sentence to previous words in a sentence in terms of their semantic content. A lot of this relies on modern progress in deep neural networks for language processing, the things that underpin Siri and Alexa and all that stuff. So we've been using those to kind of quantify, in some sense, the model the meaning of words in these stories. So, and then we had our subjects listen to it. So again, this is a very different X now that we're feeding into our analysis. It's the same EEG data. People have listened to speech, and we've modeled it using envelopes as our X, spectrograms, phonemes, and phonetic features. And when you model the data using the actual kind of meaning of words, numbers that reflect the meaning of words somehow, what we specifically did, I won't get into details too much, but what we specifically did here was we modeled how similar or dissimilar a word was to the previous words that you've just heard. So how different is the current word from its context? When you do that, you end up with a completely different temporal response function. It's much later. You know, there's nothing really happening before 200 milliseconds at all. When it comes on, it's way over parietal scalp. It's not an auditory response at all. In fact, you know, you would get the same thing if you had somebody reading this speech instead of listening to it. When you play the speech backwards, the response disappears entirely because you can't understand backward speech. There's nothing to understand. So you get this kind of all or nothing temporal response function that is entirely different from what we've been seeing before with acoustics and phonemes, because it's a different X that's you're asking a different question of. And again, thinking about this idea that sensory systems are organized in this hierarchical way, and the question that you're asking these TRFs is how does the data change as a function of changes in this particular stimulus feature? Okay, so that's just a quick whirlwind through the importance of what do you mean by a temporal response function? What is it that you're modeling? I think we're going to hear more on those ideas later in talks about encoding models in particular, and what it means to choose a different X here. But also it matters what you talk about in terms of your actual data. You know, what aspect of the EEG are you looking at? Where do you expect, you know, the stimuli or the cognitive processes that you're interested in? How do you expect them to show up in the EEG data? And what frequency bands, for example? Will they be in the phase of the data or the power of the data? And so, you know, just a couple of quick examples on that and how it can look quite different in different contexts. This was work, a line of work done by a recently graduated student from our group, Adam Bednar, who explored how the location of an object in space, auditory object, a sound in space, is reflected in EEG data. And so this axis here, this is telling us that the stimulus is moving around in space from left to right across this semicircular axis here. So this stimulus trajectory, it's not an envelope, it's a location of the object in space. And so how, if we fit a TRF now, calling, you know, negative 90 degrees maybe 0, and calling plus 90 degrees 1, and we fit a TRF to the to this variable, what does it look like? Well, it turns out that the information is carried in different frequency bands. The stimulus was moving fairly slowly, and so this figure here is somewhat reminiscent of the nice figure that Jonas had shown with the different frequency bands going on, and he had linked it nicely to the idea of kind of obligatory sensory responses that can be modulated, for example, by attention. Here we have a kind of just a fairly obligatory response that's coming from the location of the stimulus that's hitting auditory cortex from one side versus the other at different time points. And because the stimulus is moving quite slowly, it's reflected in the low frequency EEG data. However, meanwhile, over the parietal scalp, which is often related, you know, to attention and auditory spatial attention, and people are listening to this sound and trying to determine where it is. I think we had some task that they had to detect maybe, I probably shouldn't say because I can't remember, but I think it was probably detecting some gaps in the sound or something like that. So people are engaged with this stimulus. And as a result, what we saw was that in alpha power, so we've taken the power and you strip off the kind of envelope of that alpha signal, you calculate the Hilbert transform or whatever, the location of the stimulus was robustly enough encoded in the alpha power over parietal scalp. So that's just a demo of the fact that, you know, you've got to think about what, not just the X that you're sending in, but also the Y. What aspects of the EEG data might be reflecting the stimulus that you care about? What might it mean? We have followed up this initial finding to explore this question of obligatory sensory responses versus attention. And it looks a little bit less clean than we thought. I think it was the 2020 paper. Adam did a kind of a cocktail party version of multiple stimuli moving at the same time to see if attention differentially affected these two measures, but it wasn't quite as, we found that it affected both measures a little bit and not quite as separate, kind of, the dissociation between them wasn't quite what we expected. But again, anyway, for the purposes of today, the main point is to say that the Y matters too, as well as the X. Another example of that from a current member of our team, Anthony Senegal, was to look at an interesting question. And again, it was something that Jonas flagged earlier, the idea that the signals of interest may be actually happening at very high frequencies. So in electrocorticography, for example, where you record from the surface of the cortex in patients who are in surgery, especially if you're looking at things like speech over temporal cortex, the researchers using that technology almost exclusively focus on high gamma power, the power of high gamma frequencies up around 70 to 150 hertz to look for signatures of speech processing. And so we actually, Anthony asked the question, is there anything to be found in those frequencies in EEG? Because we normally assume there isn't, and we just filter it below, 30 hertz or even lower. Nonetheless, she went and had a look at it. So she did an analysis trying to reconstruct the envelope of speech using either low frequency EEG, as we had done many times before, or by taking the power of very high frequency EEG up around 70 to 150 hertz. And we didn't have tremendously high hopes for this. But what happened was as follows. The low frequency thing really worked quite well. We can reconstruct the envelope of a speech stream from your low frequency EEG really quite reliably. But it turned out we could do it using high gamma power. It wasn't great, but it worked. It was performed much, much worse. But then when you looked at individual subjects, it was really quite interesting. For example, subject 3 here, the high gamma power was kind of comparable to the low frequency. For subject 11, there was no difference between the high gamma power and the low frequency. And for subject 10, in fact, the high gamma power actually worked better than the low frequency data. So this is kind of a zoomed in version of those 3 subjects showing that high gamma power can actually add quite a lot to your ability to decode speech in individual subjects. So, the title of our paper was including measures of high gamma power can improve the decoding. Doesn't always, but again, it highlights that the Y that you're sending into your TRF analysis, is something that you should consider carefully, and it can have a big impact. Then the last thing is the actual TRF itself here in the middle, and what does it mean, and what are its limitations, what are its advantages? I think its major advantage I've already kind of talked about is the idea that you can generate these somewhat interpretable looking models that are somewhat analogous to the kind of classic event-related potentials. You can analyze the latency of them, the peaks and the troughs, the amplitudes, how they're distributed on the head. You can estimate where they're coming from in the brain, etc. And you can fit them using, relatively typical sized data sets with a reasonable level of robustness. But, it's also an assumption. We've assumed a linear model here, and it has certain shortcomings. I'll just give one quick example here of a study done by another former member of the team who asked the question, an interesting question. He had people listening to amplitude modulated noise as well as speech, just sounds that modulated in terms of their envelope. So the blue line here is the envelope. The red line is basically a different representation of the envelope, and the yellow line is just the onsets of the envelope. But what he was interested in was, harken back to the kind of a long, well-known phenomenon in auditory neuroscience, which is if you play a loud sound, you will get a large brain response, like a loud beep or click. You will get a large response that happens with kind of quite fast, short latency, large response. If you play a quieter response or a quieter stimulus, the response will tend to be smaller, but it will also tend to be later and more smeared out in time. Now, this is interesting because our temporal response function approach up till then had been to just send in the envelope and assume that the response looks the same. It's the same shape, it's the same latency. It's just bigger if the envelope is loud, and it's smaller if the envelope is small. And so that's the assumption that underlies the TRF. Show me how the data change as a function of the amplitude intensity of the speech envelope, for example, assuming that it has the same shape and it's just bigger. You know, it's twice as big when the stimulus is twice as big, and it's half the size when the stimulus is half the size, but it's got the same shape. We know from this old literature that that is very unlikely to be true. Quiet sounds, their responses are not just smaller, they're also delayed in time. So what Dennis did was he actually split the envelope up into a whole bunch of different kind of sub envelopes. Here's the loud bits of the envelope. Here's the next kind of bin of envelope loudnesses, and the next bin, and then the quietest parts of the envelope are down the bottom here. And what he did was he fit a kind of a multivariate temporal response function, one where you'll get a TRF to this top one, a different TRF to the second one, a different TRF to the next one. And lo and behold, he found what we kind of expected to find, a very large TRF for the loudest envelope sounds that was quite early, and then a smaller and smaller and smaller TRFs as you go down that got more and more and more delayed in time. So you can see that here on this kind of heat map. This B is the same data as plotted in A, but you can see that the actual peaks of the TRF are getting later and later and later as you model the response to quieter and quieter parts of the envelope. And that's also plotted here. This is the time lag. This is this plot of the red line here. So, you know, our assumption of linearity is, we know it's wrong. Here's a very simple example showing that it's wrong. When you try to actually predict EEG data using the original envelope, the blue line here, and then you do it using Dennis's approach where you actually acknowledge the fact that the TRF likely looks different for different intensities of sound, you get this purple line here, which is just, that is a very substantially improved ability to predict someone's EEG responses. The purple is really substantially higher than the blue here. So, I mean, again, I'll probably leave this, you know, for tomorrow, the kind of discussion section and stuff like that. But the idea that your EEG is literally tracking along with the broadband envelope of speech is, you know, I mean, the EEG functionally tracks along with it. But the idea that your brain is actually, there's a single bunch of neural generators in there that are tracking along with the envelope of speech makes no sense to me whatsoever. You know, there's a hierarchy in there with loads of different neurons coding for different things. And when you smush it all together, their activity goes up and down as a function of what's going on in the speech. So you can see EEG signatures that track with the envelope. But the idea that somehow our cortical activity is magically entraining to this specific broadband envelope of speech doesn't make any sense to me, which we'll leave till discussion, I think. Okay, so yeah,
am I just letting you know it's time there? Yeah,
that's time. Okay, well, the other, I can wrap up in 2 minutes. The other things we've been looking at, we've been trying to model, you know, not just how a single stimulus maps to the data and back again. You know, if you try and do stuff like, sorry, Mick, now you've cut me off after rush through your slides. But in more recent times, one of our team members has been trying to map from, you know, a whole load of stimulus features in multisensory situations to a whole load of EEG channels in a kind of a multivariate to multivariate way. And we have found that difficult to do with TRFs, albeit it's not impossible. And we have been relying on kind of sister methods like canonical correlation analysis, which share a lot of kind of fundamental similarities to temporal response functions. Again, a bit like decoding, it exploits multivariate features very well. It's a little bit harder to interpret what it all means, but Aisling has blazed a nice trail for us on kind of methods that are related to TRFs. And there's lots of others out there in terms of looking at data across subjects, inter-subject correlation and things like that, that are, again, very closely related to TRFs with complementary strengths, but also potentially some disadvantages. And then the last thing I had was just to talk about the data. You know, we started doing kind of cocktail party attention, which I think our group is reasonably well known for, and looking at the TRF itself as a dependent measure, finding interesting responses on TRFs. But this is like, Alan Power really did a tremendous amount of work. This was data from 40 subjects who each did quite a long experiment to get responses like this. The decoding approaches that we've used in the past, again, just to flag that their sensitivity on the same data set, where it took Alan 40 subjects, half an hour of data each person to find pretty TRF responses on the TRF itself. James O'Sullivan on the same data set was able to decode what was going on 1 minute single trials from individual people using this decoding framework. So again, that's just to flag that these methods have forward and backward have kind of complementary strengths. And yeah, the X matters a lot for these cognitive measures too, in terms of how attention affects them. So I'll finish up with a quick summary. The TRF, what it's doing is it's asking the question, you know, show me how the data change as a function of a particular stimulus feature. So what do you mean by a TRF? What is the feature that you care about? You get very different TRFs depending on what stimulus feature you input. Forward and backward encoding and decoding have complementary strengths. The encoding models can be used to test how brains represent sensory information at different levels of the hierarchy, visual system, auditory system, presumably for other systems too. But decoding can really be much more sensitive. Different features, different EEG features also differentially reflect the processing of the stimuli. So is it low power, is it alpha power versus delta phase, whatever it might be. And then there are obvious limitations to it. It has its major strengths in terms of interpretability and the ability to fit it, but it's limited in terms of assuming linearity, which we know to be wrong. And we've had some challenges with respect to multivariate mappings, and also with mapping from continuous EEG back to discrete variables like phonetic features, for example, or the semantic meaning of words. So again, a quick thanks to several collaborators here and various funders for all this stuff that we've been doing for quite a few years at this point. And thanks to you all for bearing with me, and I'll take any questions.
Thanks very much, Ed. That was great. So we have a few questions here. I'll start with Adam asked, is this high gamma power effect potentially driven by the frequency following response as the pitch of the voice dips in and out of that range? So he's referring to that study where you incorporated the high gamma power as well as the lower delta stuff.
I mean, that's a great question, and something we can definitely look at in terms of quantifying the kind of pitch of the speakers' voices and see how highly that correlates. Because we did look at whether the high gamma that we were getting in some of the subjects for whom we got good high gamma signals, we did try to ask the question, is that high gamma following or tracking similar to the low frequency? Are they carrying the same information? Are they reflecting the encoding of the same speech features? And I can't remember how conclusive our result was, but I think we found that they were not, that there was some evidence to suggest that the low frequency EEG tracking and the high gamma tracking were capturing somewhat complementary information about the speech. So that's definitely something we could test and look at more directly. So that's a great idea. I'm gonna make a note of that. Thanks, Adam.
Thanks, Ed. Holly Robson asked, she said, really interesting to see the application to a clinical population. Can the TRF method be used longitudinally to observe the impact of medication or therapy or deterioration in progressive conditions, or are there methodological constraints that would prevent comparison across data collection sessions?
That's a great question. Yeah, I think absolutely think it can. I think the methodological issue across different data sessions is certainly something to be careful of. But again, EEG is quite low dimensional. There's a lot of correlation across channels. So, you know, if you put a cap on and then put a cap on a year later and it's a few millimeters this way or that way, you know, that can certainly, you know, you're adding noise that you don't want to your data for sure. But it's likely to be relatively small amount of noise for things like the decoding analyses, where you're taking all the channels and reconstructing. You know, the slightly different cap placements in that situation, I think, would have even smaller effects on what's going on. So I think for sure it can be used in clinical longitudinal things. There's a lot of issues to that. I see a lot of papers these days, for example, looking at younger versus older. One common thing you see in auditory responses for younger versus older folks is larger responses for older people. And you know, it's still an open question what that actually means. I get a little nervous when I see folks writing about, oh, it's, you know, that it might be compensatory mechanisms from the older person's brain, you know, working harder to pick out speech and noise, for example. You know, I'm not sure we have evidence to support that interpretation. You know, there's likely to be biophysics changes in the skull thickness or cortical thickness or things like that that could also explain it. So there's a lot of care, but that applies to any method, I think, not just TRFs. You know, any EEG method. fMRI is, I think, you know, a lot of literature in fMRI talks about compensatory mechanisms, where in fact blood flow changes and vasculature in older people versus younger people, I think, is likely to be a very important difference in those studies, and that needs to be taken account of. But yeah, so I do think so. I think medication, we've done a little bit with, we have a grant for looking at it, kids with autism, albeit not longitudinal, and we've grants looking at schizophrenia. Again, that's not longitudinal, but you certainly can do it longitudinally, and I think testing across different sessions is certainly doable, especially using